Hello, good evening. Now we are going to see regulation. Regulation of gene expression. So we have seen certain process so far in a lot of videos. The first step is in the formation chain. The first one is the transcription. Okay. The second one is processing. The third one is once the processing over, the transportation. Transportation of what? Transportation of the mRNA from nucleus to cytoplasm. This transcription is the preliminary hnRNA. This we can say splicing. This is a precursor. Okay. Then the fourth one is translation. These are the steps we have seen. In all this, what is the expression of gene in this process? That we are going to see, right? We'll take an example from a prokaryote, bacteria, E. coli. Then we will see how this expression takes place. Okay, now can you understand? First the transcription from DNA to H and an RNA. Then what happened? I have seen, I told you, the king comes out. Isn't it? For that, the splicing, capping, tailing takes place. That is processing. After processing, then it comes out from nucleus to cytoplasm. That is transportation. Then translation. Then with the help of rRNA, tRNA and mRNA, it prepares a polypeptide chain. In all these processes, the genes are involved and it expresses and control all these activities, which we are going to see in this topic, right? Right. I will directly go into this. This is in a prokaryotic. Example, E. coli, it's a bacteria. We are seeing this in a bacteria. How it happens, right? Before going to that, the genes which are exist in the entire process of this. Okay, here in this this peptide synthesis, there is an the RNA polymerase, which does all the work in the prokaryotic. What are the work? Initiation, elongation, termination. All the three steps are done by the RNA polymerase only, right? Whereas in eukaryotic, we have seen polymerase 1, 2, 3, but here there is only one polymerase that does all the work. Okay, when these polymerase join with some associated proteins, then it will become regulatory. Then it is the promoter that become a promoter. initiation of the process takes place with the help of polymerase joined with the associated proteins. This is the first step. Okay, now let's stop here. Then we will move on to the different type of genes. Then with the diagram we will see. Okay, do you understand this? I'll give you an example now. What we are going to see is it's an enzyme called beta galactosidase. This galactosidase is controlled by a gene. Okay, clear? Now, what's the use of this enzyme? It's an enzyme controlled by a gene. And this enzyme, when lactose is present, then it converts the lactose into glucose. What is lactose? It's a disaccharide. What is glucose? It's a monosaccharide. What is a glucose? It's a source of energy. It's a source of energy, so by which bacteria gets energy. Suppose in the absence of lactose, then it cannot synthesize glucose because lactose is absent. So naturally this galactosidase is not produced because the gene is not expressing. This entire process doesn't take place. So all these process takes place only when lactose is exist. Okay, that's why in prokaryotic, when beta glucosidase acts on a lactose, it converts disaccharide into a monosaccharide. This is by a genetic professor and a biochemist. I'll tell the names. The names, okay. Written somewhere. Jacob and Jacques Monod. These two scientists together worked on this, and they have brought out how the genes are expressed while protein is synthesized. Okay, and what they found out is this system, they call it as Lac Operon. Lac is nothing but lactose operating system. Lactose operating system, that is Lac Operon. This Lac Operon contains two set of genes. One is regulatory gene, structural gene. There are three structural genes and one regulatory gene, and that one regulatory gene is expressed in the letter I. It is a regulatory gene, and here Z, Y, U. There are three genes. They are structural genes. It is a regulatory gene, and this regulatory gene codes for the mRNA, and it's a repressor. It's a repressor gene. Okay, and this Z codes for beta galactosidase, and Y permease. Permeable means allowing, that I will explain. And this is trans is an enzyme, trans acetylase. There are three genes which does control the production of three enzymes. They are enzymes. Three enzymes. And this beta galactosidase, I already told, it converts the disaccharide galactose into glucose. That is its work. And that is controlled by a gene Z. And Y controls permease. Permease is this galactosidase, once it is produced by this gene, it has to enter into the cell inside. Okay. So that permeability, that entering inside, is paved way by this gene, controlled by this gene. So it permits this to enter quickly into the cell. And the gene A, it's a transacetylase, which helps in translation process, the protein synthesis process. So by which there are four genes which is controlling the protein synthesis in prokaryotic, and that system we call Lac Operon. Why you call Lac Operon means lactose. You may ask, sir, if suppose some other amino acids, all this, triosine apron, valine apron, arginine apron, like that you can call. So the production of this is controlled by some certain set of genes. That is the regulation of gene expression done by Jacob and Jacqueminot. Okay, and there are four genes. I am going to draw the diagram, right, by which you can understand. So just try to understand. This is a repressor, this gene, and this is that gene codes for galactosidase, Y codes for permease, A codes for transacetylase. Okay. I'm just want to make the big topic into simpler one for you. Okay, right? I'll just make it. Suppose this is the first site. The gene is here. Okay, and here are our genes. Z is there, then Y is there, then A gene is there. These are the places they are operating. Okay, this is a promoter site. Suppose this I gene is a repressor. Okay, what is mRNA? I'll take. Okay, it's an mRNA. It's a repressor gene, so it produces mRNA. And this mRNA, if suppose lactose is present, lactose is absent. So presence and absence of lactose, we are going to see this expression, how the gene is controlling this activity. So one is present, this is in the absence. Another I am going to draw in the presence. Here what happens in the presence of lactose. This is in the absence of lactose. Okay, that is what we are going to see. I have just mentioned the genes. This is a regulatory. It's a regulatory gene. These three are structural genes. Okay, and what this gene does, we will see. This one produces beta this one these are the expressions of that we have seen. Okay, now we will see how it is operating, how it is functioning, right? That is what we are going to see. Oh, suppose there is a repressor. Okay, this is a repressor. This repressor will go and attach in the site adjacent to the promoter, that is called operational site. This is the operational site. When this goes and attach here, this will block that RNA polymerase function. So, no proteins. This is in the absence of lactose. This is how it is. But whereas when the lactose is present, suppose this is a repressor. Okay, when this repressor join with the lactose, suppose lactose, I am just making it in a color. This is lactose. When it joins now, this repressor, now it's becoming with this lactose. Now this goes and join at this site. This site like this. Okay, sorry, the color I will change and then it's easy for you. Now this permits this RNA to function. To start. So what does it? It prepare a proper mRNA, a protein, a polypeptide chain, a long chain is prepared. Now these three genes, so this helps in converting lactose into monosaccharide. This permits this to enter into the cell. This helps in translation. So this helps in translation. And these two helps in transcription. Transcription along with this RNA polymerase. Now when lactose join with this repressor, then it will become an operator, initiator, Okay, and this permits RNA polymerase to function. Then RNA polymerase does the work. Already we have seen these three genes are there, they produce their enzymes and they does the work. Then a long chain of protein is not produced here, but here protein is produced. Long chain of protein is produced, and this is the expression. So we can categorize this like this under see in the presence and in the absence, how it works. This is in the absence and this is in the presence of lactose. When lactose is not there, and naturally all these won't be happening. You see, all these won't be happening because already what is not there? Lactose is absent. Once it is absent, also use of producing beta galactosidase and permease, no use. So it is not produced. So RNA polymerase cannot start its function, cannot produce protein. Whereas when the lactose is there, then this gene, when lactose join with the repressor, then naturally it will become an initiation process and it initiate this site and it start the functioning. Okay, this is how the genes are expressed in a protein synthesis in a prokaryotic bacteria. Okay, we have not seen gene expression in eukaryotes, right? Very simple. So they have shown you how it is working. This the diagram is just, these are the promoter sites. The first gene is here, the three genes are here. And always this operation site is adjacent to the promoter site, so it works here. When it reach here without the lactose, the process doesn't take place. When it reach here with the lactose, then the process starts. Okay, very simple. Okay, and now we have come closer to transcription and translation. It's over. Gene code is over. Right, types of RNA is over. Okay, now we are going to see the very modern topic, human genome. How many different types of genes are present in the human body in a cell, that we are going to trace it out and to make the understand the character of human. That is going to be our next video. Okay, all the best. Bye.